Hey, hello everyone, and thank you for joining us for our webinar today, Loop-Mediated Isothermal Amplification, or LAMP, Assay Development Challenges and Solutions. My name is Beth Fry, and I'm a product manager here at Lucigen, and I'd like to welcome you all today. And before I introduce today's speaker, we just have a few housekeeping notes for you that are listed on your screen. First, please notice that all of the participants are on mute and will remain on mute for the remainder of the webinar. And just to be sure, we ask that if you are on a mobile device like a cell phone, that you mute that device just to make sure that there's no background noise. We also want to make sure that we answer all of your questions, but we are asking you to hold questions until the end of the webinar. We should have plenty of time. You'll notice on the left-hand side of your screen there's a chat window, and we ask you to use that chat function to ask questions at the end of the webinar. Also, please make sure that the little button at the bottom of the chat window is set to all. This will ensure that we can see your question and answer it. Also, during the webinar, we will be asking some interactive poll questions, which is an opportunity for you to tell us a little bit about your experience with LAMP assays. And I'll be taking over from our speaker to ask the poll questions, and you'll have an opportunity to vote live using your computer mouse, and we'll all see the results. So we hope you enjoy the polls. We think they're a fun way to make the webinar a little more interactive for everyone. Also, just a note, if you are watching the webinar in full screen mode, you'll have to exit full screen mode in order to take part in the polls and to ask questions at the end. So feel free to watch the content in full screen, but you'll have to hit escape button to exit full screen mode to interact with us. Okay, I think that's it for housekeeping notes. And so now I'm very pleased to introduce today's speaker, Dr. Dipankar Manna. Dipankar is a principal scientist here at Lucigen. He has a wealth of experience in genomics and molecular biology, along with protein biochemistry and high-throughput assay development, as well as development of in vitro diagnostic assays. And he has leading roles in developing our innovative LAMP products here at Lucigen. And we are very excited to have Dipankar presenting today's topic, Loop-Mediated Isothermal Amplification, or LAMP, Assay Development Challenges and Solutions. Go ahead and take it away, Dipankar.
Thank you, Beth, for the kind introduction. And thank you all for joining today's webinar. The topic is LAMP, Loop-Mediated Isothermal Amplification. And we will be focusing on assay development challenges and solutions. So here's the agenda. I would like to start with an introduction to the LAMP technology. Spend a few moments to compare and contrast LAMP with PCR. Next, we'll dive into detection technologies for LAMP. This is an important part to understand before we go into the next topic, which will be assay optimization. We'll talk about different factors, how important they are in the LAMP performance, and the order in which one might go about optimizing different factors. Next, we'll briefly touch upon assay troubleshooting, what may have gone wrong when things go wrong. And lastly, I'll introduce some of the Lucigen LAMP products, OmniAmp and Lava LAMP, and I'll conclude highlighting some of the LAMP discussion topics that we touched upon today, and provide you some resources you can go to. So let's dive straight into the first question, the poll question for Beth.
Okay, thank you, Mana. I will bring the poll question window over here. And the question is for you, how much experience do you have with LAMP assays? You can go ahead and vote using your computer mouse, whether you're a novice, so if you're just starting to develop your LAMP assays, if you are experienced and have developed some LAMP assays before, or if you are an expert and you have many years working with LAMP. Looks like most of our audience is just starting or has some experience. So. All right, looks like a lot of novice and experienced users, so that's great information for us. Thank you guys. We'll finish the voting here. Looks like almost everybody has voted, and there's the results. Okay, and then I'll hand it to Amanda to continue the presentation.
Thank you, Beth. Let's go to the next slide, which is introduction to the LAMP technology. Uh, the original paper, the first paper describing LAMP was in the year 2000 by Notomi et al. From ICANN Group. So it's been there for about 16 years now. It's an isothermal technology, and it's novel in that sense that it unlike PCR, can be run at a single temperature. So that's isothermal. This is not the only isothermal amplification technology. There are a few others, but certainly it's one of the popular ones. It uses a polymerase with high strand displacement activity, and there are a few of those like BST and the OmniAmp. It can be used with both DNA as well as RNA template. Uh, the DNA amplification requires the DNA pol. In case of RNA template, one needs to add reverse transcriptase or use an enzyme that has both DNA pol as well as reverse transcriptase activity, such as the OmniAmp from Lucigen. So let's talk a little bit about why LAMP. So PCR has been around longer than LAMP, and it's been in many labs, and it's easy to design primers and run the assay. But it's not a portable assay. To run PCR, you need a good instrument for cycling the temperature, which requires a significant amount of electrical power, and the assay is slow to be effective as a point of care, point of need assay. So on the other hand, LAMP being isothermal has very low equipment need. It can be run on a heat block, a battery operated instrument. The assay once developed can be affordable, sensitive, and meets most of the criteria set out by WHO for point of care, point of need assays. And the criteria acronym is ASSURED, which is as spelled out: affordable, sensitive, specific, user-friendly, robust and rapid, equipment-free, and deliverable to the end user. So that's where the main attraction for LAMP, not in the main lab, but at the point of need, near the patient, or at the farm, or if you are doing environmental monitoring, then in the field. So now that we understand why LAMP, let's talk a little bit about the next question, and Beth is going to put that across.
Okay, so our next question for our audience here is, what applications would you like to use LAMP for? Are you interested in research use only, human or animal diagnostic testing, food testing, or environmental testing? And you should be able to choose all that apply, so you can choose more than one answer if it applies to you. So looks like lots of human diagnostic testing and some research, and a decent amount of environmental testing as well. Looks like we have a wide variety of people in the audience for LAMP application. Okay, well, we'll finish voting there. Thank you very much again for the poll, for your answers, and we'll hand it back to Amanda to continue the slides.
That was a very interesting set of answers, and it's interesting to see that there are so many different ways people are using LAMP, and we hope to cover optimization for all these applications. So let's talk a little bit about the technology. LAMP uses 6 different primers, and they bind to 8 different regions of homology on target. So it's pretty complex compared to PCR, which requires 2 primers and 2 regions of homology. So the main primers are the FIP and the BIP, which are hybrid primers shown here and here. So those primers tend to be around 40 to 45 marks. The Other four primers, the F3B3, FLBL, they are about 20 mers each, the shorter oligos. And there's a good animation on the ICANN website if you're interested to see how the amplification works, because all these primers, they elongate and they kind of uh, amplify in a stepwise manner, and the animation does a good job of explaining the process. Nevertheless, let's take a quick look schematically at the process. So it can be divided into two major steps. The first step is generation of a stem loop DNA structure. This slide explains that. Uh, Starting from a single-stranded target and the FIP oligo, um, it generates a partially double-stranded template, one of which is the parent strand, the other is the nascent strand, and the F3 primer then extends and displaces the newly synthesized strand, which due to self-complementarity at the end forms a loop. This is one loop structure. The BIP then elongates from the other end, resulting in a molecule that has two loops, one at each ends. This is the key intermediate which is required for the cycling amplification. Moving on to the next step, which is the cycling amplification. The dumbbell-shaped structure or the two-loop structure is the starting point here. It can be used by the FIP to make the next intermediate, which has this structure, a partially double-stranded molecule that can take one of two pathways. In one case, the loop extends all the way to generate a fully double-stranded molecule, and another FIP can bind here and keep continuing the process, eventually generating a large concatemer of repeating elements that are inverted repeats. This molecule can also generate the complementary double-stranded, the dumbbell-shaped structure. So this and this are just complementary to each other, and this one then follow the same pathway as this one, generating another concatenameric molecule. So the end result of all this basically a large concatenamers which are inverted repeats of the amplicon. Going to the next slide. Once we understand that the amplification happens at a single temperature, it's easy to understand that workflow is simpler than PCR. It starts with sample, can be a human patient sample, can be the environmental sample, can be a food sample. No matter what, the sample has to be treated before it can be added to the reaction. LAMP being tolerant to sample matrix inhibition better than PCR, most often a simple heat lysis or heat treatment is sufficient to take the sample and put it in the assay. If it's a complex matrix, one might need to do DNA or RNA enrichment / purification, and that tends to be minimal for LAMP. So overall, this process can take 5 to 15 minutes. Once in the amplification reaction, the amplification can be done in a dedicated isothermal instrument that's portable and battery operated, or it can be done in a heat block if you are not using fluorescence detection. And most assays are rapid, so 30 minutes or less is sufficient for detection of the amplification product. Detection can be real-time or endpoint. In case of endpoint, you are looking at the end of the assay either visually or gel analysis, trying to determine amplification happened or not. If it is real time, inclusion of fluorescent dye can help, and we'll be talking a little more on that. Now that we have talked a little bit about the technology itself, what it is, and how it's easy to compare and contrast PCR with LAMP. PCR certainly requires a temperature cycling, and that's where we come across a big need for an instrument capable of cycling temperature. LAMP being isothermal, the instrument requirement is minimal. PCR requires 2 primers, LAMP requires 6. It can be done with 4, but the 2 loop primers can accelerate reaction significantly. PCR tends to be slow, taking about an hour or more for the reaction. LAMP tends to be rapid, typically less than 30 minutes. PCR also results in a less product, less than a microgram typically. LAMP can be pretty high, 10 to 20 microgram. Because of the high yield of the DNA product and the rapidity at which it forms, LAMP is suitable for or amenable to visual detection based on turbidity, and we'll talk a little more about that. PCR, due to the enzymes used like TAC and other PFU and things like that, little more sensitive to sample matrix. LAMP tend to be more tolerant to sample matrix, and this is another attraction for using LAMP, because you could do minimal sample prep and still get amplification, and thus you don't need a separate nucleic acid purification step or have complex instrument to automate that. The cons of using LAMP is that it's difficult to multiplex. PCR is easy to multiplex, so 2, 4, or even more than 10 targets can be detected in a single assay, single tube. Whereas here, because there are 6 primers and reaction is isothermal, it's difficult to multiplex, and most people don't multiplex LAMP. It's meant for 1 or 2 targets at a time. So let's jump on to detection technologies, and once we understand that, we'll realize what will be useful for optimization. There are multiple choices, and the first one, the simplest one, is we hinted at. Due to the massive amount of amplification that goes on in LAMP, there is also a byproduct of that amplification, that is inorganic pyrophosphate that accumulates in the reaction. That then reacts with magnesium in the assay, resulting in magnesium pyrophosphate. Magnesium pyrophosphate is insoluble. It precipitates out, and that's a good indicator that DNA amplification occurred. This is a good indicator for positive and negative. So a negative reaction will be clear, whereas a positive reaction will be turbid. The same property can be used for real-time monitoring of the assay if you have a simple optics that monitors turbidity. It gives you a handle to monitor real-time, but it's not as sensitive as monitoring something like fluorescence from the assay. We'll come to that in a minute. The next best or economical option is colorimetry. LAMP reaction as it progresses. In case of amplification, there's a change in pH of the assay, and if you have a pH sensitive dye, you can monitor amplification through the pH change. And so there are multiple dyes that's possible to incorporate in the assay. So that's the next easy visual monitoring of LAMP. The third one here is an agarose gel analysis. As we briefly talked about, LAMP results in concatenamic molecule, which on an agarose gel appears as ladder. These are monomer, dimer, trimer, and so on and so forth. Positive reactions tend to have this characteristic ladder pattern. A negative reaction lane will be either blank or there may be a thin smear as opposed to a ladder structure. So let's talk about the endpoint analysis a little more. Because you are looking at the end of 30 minutes or 40 minutes or 60 minutes, whatever the time may be, a positive reaction may have reached plateau at that point. So the endpoint analysis is not very informative about on the kinetics of the assay. In this example, there is a 7 log difference between the first set and the last set in terms of DNA copy number. So the amount of target that went in this reaction is 2.5 10 to the power 7 copies per reaction, and in this case it's about 2.5. So there is a 7 order of magnitude difference, but if you look at the amount of product generated, there isn't a huge difference, at least visually. And that's because the reaction has reached plateau way before that. So unless you take time point at regular intervals, you cannot infer from the endpoint how fast or how well a reaction went, or which was a more rapid versus slower reaction. And thus it's not suitable for assay optimization. The other option one has is using fluorescence. Fluorescent dyes such as Fiona green or Eva green, which is a double-stranded DNA binding dye, binds to DNA non-specifically and fluoresce. So the intensity of fluorescence is directly proportional to the amount of DNA present in the assay. So in this example, Fiona green was incorporated in the assay, and on the x-axis you are looking at that's the time scale. On the y-axis you have the relative fluorescence intensity RFU from the assay. There are 4 different conditions that were run. Condition 1 in red and condition 4 in black. What's easy to appreciate here is that if you look at the endpoint, which is about 30 minutes here, all conditions look similar, right, in terms of product output. So that doesn't help much. Whereas if you were able to monitor it real time, you can see the condition 1, the red traces, those fluorescence crossed a threshold, in this case 2000 RFU, much earlier than condition 4, which are the black traces. So the condition 1 was a rapid amplification, condition 4 was a slow amplification. And to convert this trace into quantitative numbers, we can derive a time to threshold or time to result, which is the time it took for the signal to reach or cross this signal threshold, which is 2000 RFU in this case, and call that TTR. So in this plot, we are plotting TTR with different conditions, and you can see condition 1 fared better than condition 4, and this quantitative or semi-quantitative value is useful for LAMP optimization. Let's move on to the next slide, where I bring back the gel picture, and you can see this is the endpoint assay, and this is the TTR analysis of the same reactions as we had Fiona green in each of these assays. And you can see that now we can distinguish a high target input from a low target input assay and how that affects TTR. In all these assays, we have what's called a no target control or NTC, and in that case, the signal did not cross the threshold within the detection time window of 30 minutes or so. So the TTR for NTC is 30 or greater. That brings us to our third question, poll questions, and Beth is going to pose that question.
Hey, hello everyone, and here is our third poll question for the webinar today. What type of targets do you want to detect? Are you working with DNA or RNA or both? A lot of DNA, pretty good amount of RNA targets as well. That is wonderful. Okay, so it looks like DNA is still in the lead, but there's definitely a lot of RNA folks out there as well. Okay, looks like the voting is kind of settling down here, so I think we'll finish this particular poll here, show you the results. And just a note, I see that everybody is, a lot of people anyway are asking questions and conversing over on the chat window. That's great. We will ask our speaker to weigh in on those items as well at the end of the webinar. Okay, so thank you everyone. Back to you, Manna.
Thanks, Beth. Let's jump into assay optimization now. First, what are the goals for optimization? And I have listed the top few, but it's not an exclusive list of goals. So the first one is faster TTR, that is increased reaction speed. You want to detect the positive as soon or as quickly as possible for it to be meaningful point of care, point of need assay. The second one is reduce non-specific amplification. The LAMP tend to generate more background than PCR because it requires 6 primers and it's an isothermal reaction. So one of the top goal right after the faster speed is lower background non-specific amplification. The third one is kind of related to first and second. We want the positive amplification and the background amplification to be well separated from each other. So a good separation between positive and negative amplification. And lastly, we want to detect low copies. That's certainly a top criteria, assay sensitivity. So you can add others to the list, such as assay specificity and maybe sample matrix tolerance, so on and so forth. But they all tend to fall in one of these optimization approaches. So what are the factors that we can tweak and make the assay better? So let's hear some of the list items. The top in the list are primers. As the 6 primers, and it's a complex design. Not all primer sets behave the same way. We often design 3 to 6 different sets of primers to find a good one. So design of the primers important, and there isn't an easy way to predict which one will do well. A lot of it is empirical, but certainly one can try and match TM, make sure that the AT richness and things like that are optimal. The second in the primer is the concentration of primers in the assay. That can be varied within a range. And lastly, the ratio of each primer in the mix. So typical ratio tends to be 1 to 4 to 8. 8, the most being FIP and BIP. They are the key drivers of the assay. And F3, B3 being the list which are required in the initial phase of the reaction. But one can tweak that ratio to match one's need, because it might reduce background amplification if you lower or change some of those. The next in the list is enzyme. Concentration of enzyme can have a significant impact on the assay, and we can tweak that. Reaction temperature is a big factor. Depending on the primer set, the assay may have different optimal temperature. It does depend on the enzyme also. Magnesium ion is a key factor, and within a range we can tweak that, and that also depends on the primer set target combination. Reaction pH is not a favorite factor to optimize, but it can impact, and we'll have some examples of that. Lastly, PCR additives such as betaine and trehalose, those can improve LAMP assays too. And again, one has to figure out impact of those in a specific assay, and whether it's worth the trouble to add another reagent to get a little more benefit out of, speed out of the reaction. Design of primers. So in this example, we are looking at 7 different primer sets for the same target. So as you can see, primer set 1 to 7, they have different TTR coming from the same target, and set 1, 2, 3 is much faster than set 4 through 7. And so this is a typical success rate, maybe 50% or lower fraction of the set you design are good for the specific target you're working on. And in this case, we have all these sets run at 3 different temperatures, and the trend continues, but at higher temperature, the reaction is a little faster. So once we decide which set to work on, we can characterize it in a little more detail. And in the next slide, set design 1 was used to look at assay sensitivity, and in this case, a target was diluted serially, and we have a no target control. The reaction is pretty rapid, under 5-minute TTR for high target concentration, and about 15-minute TTR for a low target concentration. And the background stayed low, that is undetectable, up to 40 minutes. Let's look at primer concentration. That's certainly something one can tweak. The typical concentration is written here, FIP and BIP at 1.6 micromolar, and F3B3 being the least, 0.2 micromolar each. And on the left hand side, the orange bars are amplification from a positive target, and the gray bars to the right are background amplification. And as you can see, we are varying primer input from the typical 1x to all the way down to 0.1x. As you lower primer concentration, the positive target stays where it is up to a certain concentration, and then it slows down. The background also slows down, so it's a balance between the speed requirement from positive and the background, and the best concentration in this case tends to be is around 0.2x primer concentration, where we can detect the positive pretty rapidly, and the background is at least 50 minutes or higher. And although the assays are run for 50 minutes in this case, in a typical, once you have developed it, you won't be running it this long. You may be running it at up to 30 minutes max. And so this is useful in knowing that your background stays low till at least 40 or 50 minutes, so that there is no overlap between the positive and the negative amplification. Going to the next slide, effect of enzyme concentration. Certainly, higher the enzyme, faster the reaction up to a point, but that runs the risk of higher background amplification too. So in this case, the enzyme concentration is varied on the x-axis. This is the 1x, the rate concentration. If we add more, the positive doesn't really speed up that much, but you run the risk of the background coming up. If you lower the enzyme, things may slow down, but you also push the background out. So 0.8x may be a good balance here, where you can detect the positive reasonably well and not have the background amplification coming up within your detection window. And it can be different for different assays. Next important factor is the temperature of the assay, and each primer set you design can behave differently, partly because of the TM differences of the oligos. And in this case, a single primer set was assayed at various temperatures from 66 to 72 Celsius. There are three positive target levels and one NTC. And you can see that at 72, the assay is rapid and robust. We can detect the positives under between 10 and 15 minutes, whereas the negative tends to stay out at least up to 50 minutes or longer. So this is a good condition. If we lower the temperature, things tend to slow down, and at 66, we don't see any amplification even from the positives. Um, this is an extreme example where the 72 Celsius looks the best. There are other cases where maybe 68 or 69 may look better. And this is using our OmniAmp enzyme, which has a temperature optimum of 68 to 70, 66 to 72. Magnesium, a very important factor in the assay. All DNA polls require magnesium to do amplification, polymerization. So certainly we need magnesium in the assay. At high magnesium concentration, assay tends to run faster. These are two positive template. The gray bar is the no target control. So you can see at high, even though the positives are fast, the background is also fast. So if we lower the magnesium, we tend to push the background out. So in this case, 6 millimolar magnesium may be optimal because we can detect the positives still within 30 minutes and no background amplification even up to 60 minutes. We need a good separation or good distance between the positive and the negative so that there is no false positive in the assay. Let's move to the next slide. We're talking about now pH. pH is largely dependent on the enzyme, and so it can be optimized to work with a specific enzyme and then keep it constant. So in this example, the pH of the assay has been varied from 7.8 to 8.8. The blue bars represent positive amplification from positive target. That's not affected significantly. The red bars are the no target control, the background amplification, and that changes to some extent with increasing pH. And in the slide next, it's much clearer. We are looking at background amplification only in this gel, which is a smear pattern. At low pH, you see the background amplification, and it goes away at high pH. And you can monitor the same thing using fluorescence, in which case the red traces are positive target, the black traces are the no target control, and at low pH, pH 8, the background amplification is visible. At higher pH, it disappears. So pH can be a player in the assay. Lastly, but not the least, additives such as betaine can improve assay performance. In this case, the orange bars are the positive target and the gray bars are the no target control. Without betaine, the positive comes up under 20 minutes and the negative beyond 30, but the separation is less than ideal. We'd like to have a better separation between positive and negative, and so if we increase the betaine concentration at 400 millimolar, we can see the positive has sped up and the negative has slowed down, and we have a improved delta TTR between positive and negative. And many times additives does help in lowering background, but cannot make an assay work if it didn't work without the additive to begin with. So it can improve, but it doesn't dramatically change the outcome. So the additives tend to be the last choice of optimizing the assay. Now, having gone through all the factors that one can optimize, it does look overwhelming, but honestly, there are a few that are top priority, primers being one, magnesium being second, temperature being third, and so on. But if one is really adventurous and want to optimize a lot of different factors, DOE, design of experiment, might be the path to go. That is a statistical analysis that helps to crunch data, and the flow chart here shows the way for DOE. One needs to set the objective, in this case reduce TTR for positive and increase the TTR for the negative. And then we need to choose what are the factors you are trying to optimize. In general, you can include all the factors we talked about, but you may deal with the major ones one at a time, such as temperature, and get it out of the way, and deal with the salts and additives in the DOE, such as magnesium, KCl, ammonium sulfate, and so on. DOE requires a quantitative response output. In this case, that's TTR, the time to result. You can design either a screening design or for first picking out which has the biggest impact on the assay, or you can choose a response surface design where you are trying to look at interaction of multiple factors and optimizing all the factors together based on your comfort level and the goal. The next step is the strategy, deciding on the target level, replicate number, etc., run number of runs. Once you have the data, it's fit into the software to generate a model and analyze the model. And last but not the least is confirming the model that you have the optimal parameters for the assay. So DOE can be helpful in optimizing a variety of factors together, and it is better than one factor at a time in some cases. One more thing, assay requires a sample input, and the sample can affect the assay for sure. So crude sample, although tolerated, may require some pre-treatment. In this case, we are looking at untreated stool sample, which is in the blue bar. Stool sample needs to be diluted in some buffer before it can go in the assay. But if that dilution is untreated, you may not see any difference between the high target input in the sample from the no target control, the blue bars. So they all have about similar TTR. Whereas if you treat the sample minimally, even just a heat treatment, 90 C for 5 minutes, you can make out that target at high, medium, and even low can be distinguishable from the no target control. So heat treatment of the sample may be required and may be sufficient in some cases as sample prep for LAMP. We do not need to purify the nucleic acid in all cases. So now that we have gone through the major factors that can be optimized and talk a little bit about assay optimization, let's talk about troubleshooting. When things fail, what to look for. So primer design being the most important factor, certainly if your primer assay is not working, you have to look at the primer design. Maybe design a new set and see if that works. Sometime maybe a primer set was working and then you got a new batch of primer and things stopped working. And we have seen that happen often. So primer purity is important, particularly because the FIP and BIP primers are pretty long primers. They are 40 mer plus. So HPLC purification for FIP and BIP will help getting consistent result. Otherwise, it might vary from batch to batch. Sample matrix, all sample matrix are not created equal. So even though you may have gotten good result from one particular urine sample, it may differ with another urine sample. So if things don't work out, that makes sure that your sample matrix is not the factor. Reagent, certainly, making sure that the quality and the stability of the reagents are not an issue. And if you're adding crude sample to the assay, making sure that it's not altering the salt balance and the pH of the assay significantly certainly will help. Lastly, I have highlighted in bold the target contamination issue. It's often not well appreciated that LAMP generates a huge amount of product, much more than PCR. So if we do take the amplified product downstream for downstream analysis, like gel, running agarose gel, we handle it. We run the risk of contaminating lab space and lab equipment like pipette man with that amplicon, and that can give you false positive amplification, which is difficult to clean up. So certainly one needs to be aware of it, and the best practice will be to keep the workspace where you are setting up reaction separate from analysis space where you are running agarose gel and handling the amplicons. But it is a very big issue, and one certainly needs to be mindful of handling the amplicons after amplification, and if at all possible, not to open up the tube at all. Let me introduce a couple of products that Lucigen offers for carrying out LAMP. One is OmniAmp. It's a kit for doing DNA LAMP as well as RNA LAMP, and we are in the process of developing a second product called Lava LAMP. It will be a LAMP master mix where you have all the reagents included in the mix. OmniAmp uses a thermostable polymerase that was discovered at the Yellowstone hot spring by Lucigen scientists. So it's a very unique enzyme, has DNA polymerase activity, but also has reverse transcriptase activity and a strong strand displacement activity. So the enzyme is suitable for DNA as well as RNA LAMP, and the kit is called OmniAmp. Here are some of the features of the OmniAmp polymerase. It's highly thermostable with a strong strand displacement activity, single enzyme suited for RNA and DNA detection, isothermal amplification, can be stably dried, can be freeze dried or even air dried, and is highly tolerant to sample inhibition. We have used this enzyme successfully developing human diagnostics, animal diagnostics, viral, bacterial, fungal detection kits, as well as environmental detection kits. So the enzyme has been tried and tested in the field in many different applications. We are in the final stage of developing a master mix called Lava Lamp. It's coming soon. It's in the convenient 2x master mix format. It's really sensitive, specific, and rapid. It's tolerant to crude sample. It can be heated to 90 C for target denaturation. I briefly saw someone mentioning target denaturation in the chat window. Yeah, target denaturation is not required, but does help to improve sensitivity of the assay. And in this case, one can add the target to the master mix and heat the complete mix to 90 Celsius for target denaturation. It will be in two different formats, one with the fluorescent dye included and one without for ease of use. And the master mix is formulated such that the mix itself can be freeze dried for ambient storage. That brings us to our last question, and Beth, take away.
Okay, so last question. We would like to know what you would like to see in a LAMP master mix. What is important to you? Let me just get the window up here so that you can see. So amplification yield, and please select your top 3 if you can out of this list. So yield, time to result, sensitivity, reproducibility, no background or low background, time difference between the positive and the background signal is the total time that you have for your assay window, cost per reaction, ability to preheat the complete reaction without inhibiting amplification, so that might be important for people that are worried about sample prep, and lyophilization compatibility. Looks like sensitivity and reproducibility are pretty big concerns, or pretty big needs, I guess, for people, as well as background level and lyophilization. So there's a little bit of people who would like some lyophilizable enzymes as well. So, okay, well, we'll go ahead and finish voting, and we'll let finish up his slides here, and then we'll get to some of your questions. Thank you.
Thank you, Beth. Let's conclude the webinar by going over a few of the major points you talked about today. So it's a very interesting technology, particularly suited for point of care, point of need assays, and it's sensitive. It can be as sensitive as PCR. It's highly specific and rapid. Requires 6 primer, which makes it a little more challenging than PCR to develop, but certainly can be developed to make a robust assay. There are few factors that one can optimize, which include the enzyme concentration, primer design, magnesium, temperature, and so on. Optimization can be done one at a time, one parameter at a time, and in that case you need to prioritize which ones are the top ones, or it can be done using DOE, where you are throwing in multiple factors in one part. Lastly, it is a very attractive technology, as I just mentioned again. Once developed, the assay is very portable, and we have developed assays that that has been deployed in the field to detect invasive species, for one example. So it's very attractive as a platform, particularly for point of care, point of need, and there are FDA cleared human diagnostics that uses LAMP. So the application-wise, the field is wide open and very broad. Thank you for your attention. And, I would like to mention a couple of resources. The LAMP primer design website from ICANN is the second one. The LAMP primer design software from Premier Biosoft is the third one. Our OmniAmp publication link is available here, but we are always available to you through our tech support team. We have an awesome tech support team. So if you have question and if your question is not answered today in the chat or the webinar today, please feel free to reach us, and we'll be happy to help you with your LAMP development project. Thank you again for your attention.
Now we're going to move on to some of the questions. There's a lot of great chat here. I really think it's awesome how engaged this audience is. This is fantastic. So I want to try and get to some of these here. William Taylor asks, is it possible to get only one product from the reaction rather than a smear? Dipankar.
Sure. If you remember the first few slides, the LAMP by virtue generates a concatenameric molecule, and even if you are looking at the endpoint assay, you get different mers. So you get monomer, dimer, trimer, decamer, and so on and so forth. So if you run it on a gel, you will see a ladder of different length products. But if you digest it with a restriction enzyme, if you know the amplicon and you know the restriction sites, use the right restriction enzyme, you can cut that concatemer into a single length product. So all those ladder will then converge or merge into a single product. And that can be a typical diagnostic feature of your assay, that did you generate the right product or not. So it can give you a single band if you have the right restriction digestion of the product.
Okay, great, thank you. Next question I'd like to get to is, do you need to denature the DNA template for the first step to let the outside primers bind to the template?
That's a great question. We have run a lot of assays both for DNA and RNA, and we see that denaturation is not required, but it can help improve the sensitivity of the assay. So if you do a denaturation and then add it to the assay, or add to the assay for the lava lamp and denature it, you get a better sensitivity, maybe half a log or somewhere in that range.
Okay, great. There's a couple of these later questions that we want to get to too. So for the presenter, so can OmniAmp be heated at 90 degrees or above? Original papers and protocols from the website, I assume that means the Lucigen website, seem to differ. Do you have any comments on that?
OmniAmp is not formulated to be heated to 90 Celsius. It is formulated to work at the lamp temperature, which is around 68. But the lava lamp product, which is going to be coming out soon, is formulated to be heated, can be heated to 90 for target denaturation. So we are right now in the final stage of that development. So it will be out soon, and that one you can hit it to 90 for 5 minutes.
I also have a question. How does the freeze drying procedure, how does drying with primers or drying primers separately affect the background as far as false positives?
Sure. So we have done a lot of air drying, and we also have done a lot of freeze drying, lyophilization. In case of lyophilization, you can lyophilize the master mix or your own developed mix with our enzyme, it works fine. And with this particular enzyme, it is stable. And then add the primer at the time of the assay. You can also lyophilize with the primers. And we haven't seen any significant difference between the two. So that is, you can freeze dry with or without the primers. In case of air drying, you want to leave the primer out if you are air drying, because air drying, as things are drying, things tend to concentrate, and that can change the assay kinetic. And so you will not get the performance you're looking for if you air dry with primers. But we have air dried without the primer and then added it later. In that case, it works.
Okay, great. And then one more question here. Does lyophilization affect the enzyme activity for LAMP or RT LAMP?
Not to our enzyme. So OmniAmp and Lava LAMP, they are both suitable for lyophilization. And we have developed quite a few products where it's lyophilized in single-use aliquot with the primers. And both the weight reaction, the control, and the lyophilized reaction behave identically in terms of positive detection, sensitivity, and the background amplification.
Okay, and we're a little bit over the hour, but there's I want to get to. We'll do one more question. Since LAMP primers are generally based around design for 65-degree reaction temperatures, how do the design tools work for primers with OmniAmp, which has an optimally higher temperature near 70 degrees?
Certainly, you can change the design to make the TM a little higher, and I have done that even with the PingBiosoft software. You could have the TM as an input criteria, or you could add additional bases, but that's one way. But at the same time, it's also worth mentioning that we have taken a lot of published primers that were originally designed for BST enzyme and put it with our OmniAmp, and all of them have worked to various degrees, and we don't need to specifically design it for OmniAmp, but it will be better if you have the handle.
Okay. Thank you. I guess we're a little bit over the hour, so I think that's about all the time we have for today. I'd like to thank our speaker,